So folks, welcome. I had recorded yesterday's lecture and I looked at it and it was pretty bad. So I'm trying a different approach, you guys. I'm trying to angle it so maybe you can sort of see the slides on the Prions PowerPoint. I was referring to the slides a lot, so I'm going to try it again, you guys, today and see if this works. Folks, just business. We will have Open Lab Friday from 8 a.m. to 10 a.m. And folks, do remember Monday is our lab exam too. And it's also the day you're going to be turning in your lab study guide packets and lab results. So I think you all got a checklist. So you guys, with regard to the lab study guide packets, if you have results or notes or anything, you're not sure if you should turn them in or not, turn them in, right? Because the more you turn in, the more points you're likely to gather. So if in doubt, just staple, any additional notes to your lab study guide packets. Okay, and then folks, let's see here. Our approach today is we're going to finish Prions and then start the next unit on medical microbiology. And folks, there's this blue single-page handout, double-page handout here. This is a worksheet to help you get ready for some of the medical microbiology unit questions on lecture exam 3. And I call it a worksheet and homework. You'll see on one side there's many fill-in-the-blank type questions. These are with regard to virulence factors, substances, structures that microbes make that help them cause infectious disease. And so these would be really yummy lecture exam 3 type questions. And then, folks, on the opposite side, this is absolutely going to be a short answer question on lecture exam 3. This is a table comparing and contrasting bacterial exotoxins to bacterial endotoxins. And we will, folks, we will cover this. It'll probably be next week, but it's a lot of information, so I wanted you to have the worksheet and the table so you could start working on it early, right? So you won't have to try to cram it at the last minute. Okay, and then folks, just so you know, and I know you guys are probably tired of me giving you too much information, but talking about UV radiation and damage and finding dimers and how it can increase our risk for cancer. Well, I've had a basal cell carcinoma removed from the upper part of my lip, and maybe I got another one. So they did a biopsy yesterday. That's why I've got this old bandage here. And my brother, who's hilarious, he says, I said, well, I'm going to look kind of funny, when I go to lecture. And he says, just tell your colleagues you're in a bar fight. I go, oh, that will go over well on my reviews, right? But anyway, you guys, so that's just what's going on with this. Okay, so folks, last time on the Prion PowerPoint, I realized I pulled up a previous version of it, and I apologize. I had updated it a little bit. So I just wanted to hopefully get back on track on the right Prion PowerPoint. So folks, I think most of the slides were the same, but this was one that I wanted to show you because this is a cartoon showing where on cells of the nervous system prions can be found. And again, folks, I still have so much to learn about prions, about normal function and how they go through this process of causing neuron damage. But you guys, this is what they call a presynaptic neuron, and this is a postsynaptic neuron. And folks, the cartoon, I should be doing it like this. The cartoon for the prions, you guys, this is their cartoon for the cellular prion right here. It looks like a balloon with a tail on it. So you can see we can have possibly prions in these vesicles. And my guess is, you guys, maybe the vesicles are containing neurotransmitters. And then furthermore, you guys, we go down here, and we can see these prions on the surface of the neurons as well. Here, over here, and over here as well. So again, you guys, just emphasizing that in all of us right now, we have good normal cellular prions associated with cells of the nervous system. And then what we also want to remember is in the ingestive form of prion diseases, our white blood cells can also carry prions, and prions can also be found in lymphoid tissue, and we'll come back to that. Okay. Now, folks, help me out here. We've gone over the kind of our model for how the prions misfold and then form filaments and then stack and somehow trigger the neuron cell death, right? We've gone through this already. Okay, good. And then we said the mechanism by which the misfolded prions trigger our neurons to undergo the apoptosis, the programmed cell death. I don't think it's totally understood yet. So much mystery associated with the prion diseases. And this, folks, I really wanted to emphasize this, that our immune system does not recognize the misfolded prions as being foreign. And this is bad news because it means our immune systems won't mount an immune response against the misfolded prions. So that's bad for us for two reasons. One is, right, there's no immune response to try to protect us from the misfolded prions, maybe try to eliminate them. And the second reason this is bad is that this means we don't make any antibodies against the misfolded prions. And not only could the antibodies be protective, but we use antibodies, you guys, to help diagnose whether a person or animal has been infected with a pathogen, right? Most pathogens our immune system see as foreign. We'll make antibodies against them. So we can look for those antibodies against the pathogens to diagnose a human or an animal that might still appear healthy, to diagnose that they've been infected. And that's really what we need with these prion diseases. We need easy blood tests that would let us know, is this animal infected with a misfolded prion? Is this human perhaps incubating a misfolded prion? That would be really good before we do medical procedures to know if our patients maybe are incubating a prion disease, right? So this is really, truly bad news for us that we don't make any antibodies against the prions. Yeah. And then, folks, and I so apologize, had we gone through the short history, like that scrapie and sheep and goats, good. And then Creutzfeldt-Jacobs diagnosing the neurological disease, and then the Kuru and the Maori people, Papua New Guinea. And, folks, had we gotten through just our little tip? Okay. So, you guys, with regard to the prion disease which causes the disease known as Kuru, and this is, really heartbreaking story. Kuru was identified in the 1950s in the Foray people of Papua New Guinea. And probably, folks, what happened was one of the elder members of the tribe had developed a sporadic form of Creutzfeldt-Jacobs disease. And the sporadic form occurs in us old people when our prions spontaneously misfold, right? We still haven't figured out what the triggers are. But it was possible that one of the elders in the tribe had died of the classic sporadic form of Creutzfeldt-Jacobs disease. But amongst the Foray people, one way that you honor the deceased person is that the women prepare the body, and then the body is cooked and then consumed, right? So this is called mortuary cannibalism. And because it's really different from our culture, folks, a lot of people are like, ooh, that's gross. But you guys, a lot of us, our cultures have us do things that other cultures would find really disturbing, right? And for me, I feel like I understand this. Like, you have somebody that you loved has died, and in the act of consuming their body, to me, I can see it's like, yeah, you're trying to maintain their spirit with you, right? But you guys, this had devastating consequences, because when the first elder died of Creutzfeldt-Jacobs disease and the body was prepared for consumption, the disease causing prions then were passed on to all the members of the family, of the tribe. And again, folks, we want to remember the misfolded prions, cooking will not inactivate them. And furthermore, it's thought, because the women were responsible for preparing the body, they would be at really high risk for maybe cutting themselves and coming into contact with tissues or fluids that contain the disease-causing prions. And folks, my understanding is that the men of the family of the tribe, they were more likely to have maybe eat muscle, right? And the women and children were more likely to eat maybe the brain and intestinal tract. And certainly, you guys, in Creutzfeldt-Jacobs disease, the brain is chock full of infectious prions. And then later in the ingestive form, the intestinal tract, the lymphoid tissue, is chock full of prions. And as a result, then, through mortuary cannibalism, the disease-causing prions were spread throughout the tribe. And it was absolutely devastating. In the Australian documentary, folks, there's a link to it. It's called Kuru, the Science and Sorcery. Incredible documentary. They interview one of the Foray elders, and he was saying at one time there was almost no women left. Like, all the women had died, and a lot of the children were dying too. So once the mortuary cannibalism was halted, then the number of Kuru cases went down dramatically. And again, folks, Australian documentary is just phenomenal, but truly heartbreaking. So if you're not in a good space, don't watch it because it's really heartbreaking. But they also make links to other prion diseases that we're dealing with today, such as the mad cow disease and the variant form of Creutzfeldt-Jacobs disease that is transmitted through eating beef that has the BSE prions in it. So it makes some links to still what's going on today. Oh, and folks, this is the link to that outstanding Australian documentary. Yeah, it's just phenomenal. But and I think I've shared this with you guys. Every time I watch it, I cry. It's just, oh my God, but very powerful, very powerful. So again, you guys, if we're talking, now we're not talking Kuru, we're talking about another form of human prion disease, the Creutzfeldt-Jacobs disease. So the frequency is about 1 case of Creutzfeldt-Jacobs disease in humans for every million. So it's not very frequent, but it sure is devastating. And there's 4 basic types. There's the inherited or familial form. And this is where you have your gene for the prion protein encodes an amino acid sequence that makes misfolding highly likely. So this is devastating. This would be passed down generation from generation. And it's tough, you guys, because this is called autosomal dominant. And that means if you inherit one of the disease prion genes from one parent, you are going to develop the disease, right? All you need is one gene from one parent. And then, folks, the second form is called iatrogenic, and literally iatrogenic means caused by the doctor. And what we mean by iatrogenic is these are cases where humans become infected with the CJD prion through medical procedures or from receiving, say, transplanted tissues or transplanted organs. And again, you guys, the problem is with CJD, there can be a long incubation period. So I could be incubating CJD right now, and although you could argue, well, she is starting to show clinical signs and symptoms. Anyway, all right, you might not say, okay, it isn't obvious that you're infected with CJD, right? But if any medical procedure occurs, if like I'm going to be a donor when I die, I want my organs and tissues donated, right? Any of those tissues or organs that have the disease-causing prions then will be passed to the recipient, right? And again, you guys remember we don't have like an antibody test to see if I'm positive or not. Also, back in the day where they had not yet done genetic engineering to have human growth hormone produced through genetic engineering cloning. In the old days, if children weren't growing well, one therapy was to take a pituitary extract from cadavers from people that had died and inject the children with the pituitary extract because that would have the human growth hormone in it. But we now know that some of those pituitary extracts were contaminated with CJD prions. So those kids got infected. Yeah, so truly a nightmare. Corneal transplants, folks, have been contaminated and caused transfer to corneal transplant recipients. And also dura mater transplants. The dura mater, you guys in A&P, you know, it's that tough covering on the brain and spinal cord. So like a donor, if somebody dies, they could donate their dura mater for maybe somebody that's been in an accident or maybe I don't know, cancer or something. And the dura mater can also be infected with the CJD prions. But by far, you guys, the majority of CJD is this sporadic form. And again, we want to stress this occurs in old people. 50 years of age or older, and it's called sporadic because for some reasons we can't quite figure out when we age, our prions might start misfolding what we think spontaneously. It might be their triggers, and we just don't know the triggers yet, but the majority of CJD cases, again, are considered sporadic. But again, this is an old person's disease, right? Not in young people. And the reason I wanted to stress that was it was because in Britain, I think this was back in the 90s, young people your age started presenting with signs and symptoms that sounded just like Creutzfeldt-Jacobs disease. But the healthcare providers knew something was wrong, and what was wrong? What was wrong, you guys? You're too young to get Creutzfeldt-Jacobs disease, right? You're way too young. And so eventually people tried to figure out what was going on, and you guys know that, you know, the nightmare of this. It was discovered that this variant little v, variant Creutzfeldt-Jacobs disease. It was an infectious form, and the way the young people were getting infected was by consuming beef that was contaminated with a mad cow prion, thus showing that mad cow disease, BSE, bovine spongiform encephalopathy, is a zoonosis. What do we mean by zoonosis? Yeah, a pathogen that can be spread from a non-human animal to human, right? And did I share with you guys the British agricultural minister when mad cow disease was first identified in Britain? And people were saying, is it safe to eat the beef? He said, oh, no problem, right? And ate this great big hamburger made from British beef. But it was less than 10 years later that the young people started showing up with it. It's just, man, oh man. Okay. And folks, people ask, well, if I eat beef and I'm worried about BSE prions, what if I cook my meat really well? Is that going to do it? Nope. No. So you can cook your beef as thoroughly as you want, as well done as you want, and that is not going to inactivate the prion, right? So bad news. And folks, this was me just getting nerdy. I was just searching for tables. For those of you that are interested, I wouldn't ask this on the exam, you guys, but it's just like comparing the different types of Creutzfeldt-Jacobs disease. So they're just telling us how it's acquired, so basically transmission, average age of onset, and initial symptoms. And it is fascinating, you guys, to me, and again, I wouldn't ask this on the exam, but it appears that the different human prion diseases, the prions hit different parts of the brain, and therefore you might have different clinical signs and symptoms depending on which parts of the brain are being affected. And to me, I don't know why this is fascinating to me, but it is. It's really fascinating. But again, folks, you don't have to worry about that, which parts of the brain are getting impacted. This, to me, folks, is a little bit troubling. So this is a graph showing sporadic Creutzfeldt-Jacobs disease in the UK. And you guys, what's happening as time is progressing? We're seeing an increase in sporadic Creutzfeldt-Jacobs disease. And I would argue, you guys, I think some of those sporadic forms are which form, actually? The variant form, yeah? Because the reason is we know that prions, incubation period for prion diseases are often long, years and years. Incubation meaning we're infected, but we don't have clinical signs and symptoms yet. And I'm wondering if this increase is not an increase in true sporadic, but this is variant Creutzfeldt-Jacobs disease in which the people were maybe infected, in the 80s or 90s, and they're only now developing clinical signs and symptoms as they're older. So you guys, Britain went through an eradication campaign. So what they did is any cow, bull, steer, calf that was identified with mad cow disease with BSE, that animal would be slaughtered. The entire herd would be slaughtered. And if the animal had been born in another flock, often they would track back and eradicate that herd as well. So, I mean, this was horrific. But the reason for that was that they wanted to try to eliminate the BSE prions in beef. So, right, it's not spreading to humans. So this to me is like, well, why are the cases still going up? So to me, it's like long incubation periods, but also I think some of those cases could be variant Creutzfeldt-Jacobs disease. Yeah. All right. And just some more tidbits, you guys. We probably hit this already. So we wonder like, well, what's my chance, of developing Creutzfeldt-Jacobs disease? And it turns out, you guys, it seems like it is a risk factor if in the amino acid sequence of your cellular prion, you have at position 129 a methionine. That appears to be a risk factor, but you guys, you don't need to panic because almost half of us have the methionine at position 129, and half of us don't develop Creutzfeldt-Jacobs disease. So this might be a lot like cancer, you guys. It's multifactorial. There's numerous hits going on. There's some genetic predisposition, but maybe there's some environmental hits going on as well. We just don't know. We just don't understand it. Yeah. And again, those of you going into dentistry or medicine, and I think just the public, you guys, we need to be aware of this. So we want to remember the prions are some of the most resistant biological structures. disease-causing prions in meat, cooking won't do it. Will normal autoclaving inactivate you guys, the disease-causing prions? No. And what's normal autoclaving again, folks? What are the conditions? 121 degrees Celsius, 15 pounds square pressure over atmospheric pressure, and depending on solids or liquids, 15 to 20 minutes, right? That'll destroy endospores, it'll destroy all cellular organisms, viruses, but it won't destroy the disease-causing prions, right? We know, folks, that in cattle, in sheep, goats, and we'll see also that deer, moose, and elk can also have prion diseases. We can find the prions in the muscle, the meat, in the lymphoid tissue. It's also in the blood, folks, so we need to worry about blood transfusions. As we said, UV won't destroy the prions, normal autoclaving won't. Of huge concern in hospitals and probably dental offices too, you guys, in that if you do a procedure on a person incubating a prion disease, right, and the surgical or dental instruments are contaminated, excuse me, of course they're going to be cleaned and autoclaved, but it is theoretically possible if there's any misfolded prions, those instruments, when used again, could perhaps introduce the misfolded prions into the next patient. And that's, God, what a nightmare that is. Your colleagues have shared with me, your colleagues that work in surgical units, that if any kind of medical procedure is done on a patient with a known prion disease, the instruments are incinerated. They don't even try to, decontaminate them and reuse them. And again, folks, of huge concern is that in tissue or organ donors, right, if we don't know we're incubating the prion disease, we won't know the tissue, the organ is prion positive, and so then we pass it to the recipient. And again, folks, we need to be aware that prions can be carried by white blood cells, so even blood, right, can be a source of them. And indeed, you guys, maybe some of you know this, but if you have lived in Britain, I think from the onset of the mad cow disease through today, like here in the United States, like if you go to donate blood, they won't take your blood, and it's because they're worried you might be incubating a prion disease, right? So I lived in Scotland for a year right before the outbreak, so I might be in that bunch where they wouldn't accept my blood and maybe not accept my organs now, because living in Britain it was like the year preceding the mad cow outbreak. And folks, you don't have to memorize this, but we're always, we're so worried about, medical instruments being contaminated. So much research has gone into, how can we decontaminate? How can we inactivate disease-causing prions? So they've come up with these really harsh protocols of like soaking instruments in sodium hydroxide or bleach, autoclaving, twice. But you guys, a lot of medical instruments are so delicate, they can't survive that kind of treatment, right? So that's the concern. What about like endoscopes, right? Endoscopes have become notorious for even transferring, when you're doing the wonderful colonoscopies, right? If the endoscopes don't get cleaned properly, they've shown that the contaminated endoscopes can transfer antibiotic-resistant bacteria from one patient to the next. Well, imagine with prions, right? Ugh, it makes me not want to go get my colonoscopy. Anyway, so you folks, you don't have to memorize this, but it's just be aware there are these very harsh treatments that can inactivate the disease-causing prions, but a lot of medical instruments can't survive this kind of treatment. So incineration will destroy the prions, right? And indeed, you guys, going back to the mad cow outbreak, remember how we said the British government did eradication campaign? Normally, if you have a farm animal and the farm animal dies on your farm, like one common thing was when you just dig a big hole in the farm somewhere and bury the animal. But because we know those prions in that carcass can remain infectious for years and decades, who knows how long, it was decided that all the cattle that were killed, they would be incinerated. And I can't imagine, you guys, how horrific that whole process must have been of going in and killing all the animals and then incinerating them. I'm just like, psychologically, I think the impact would have been horrific for the farming families and the people involved. Now, in the European Union, folks, it might be they're a little bit ahead of us because in the EU, they've approved an enzyme that can help remove the disease-causing prions from medical instruments, but it hasn't been approved here in the United States. So, yeah, well, yes, okay. And again, folks, the tough thing is trying to diagnose these prion diseases, early on. So with animals and humans both, one of the clinical signs is behavior changes, psychiatric changes, inability to balance, like being off balance. Classically, historically, the way the diagnosis has been made is you wait for the animal to die, or the human to die, and then you take thin sections through the brain and stain them and look at them under the light microscope to look for those holes in the brain, right? But that's after the person or animal has died. We need what's called an ante-mortem diagnostic test. Before the human or animal dies, we need to know, are they infected? With animals, you guys, what they are doing is, like in cattle, they'll do a tonsil biopsy. And it's because in the ingestive form, they've discovered that often lymphoid tissues like tonsils will be disease-causing prion positive. So that is tricky, though, trying to do a tonsil biopsy on a big cow or steer. That's also a little tricky, but at least you can screen them to see if they're prion positive. And then, folks, here in the United States, we've got a scrapie eradication campaign trying to get rid of it out of our sheep and goat flocks. And what they'll do is they'll do rectal biopsies, ouch, and they'll also do a third eyelid biopsy, right? And again, these are living animals trying to screen them, right, to see if they might be carrying the scrapie prion. But again, you guys, it'd be so much easier if we made antibodies against the prions, right? We could just screen for antibodies, but our bodies don't make antibodies. And again, folks, this was me just trying to update this. I was like, haven't we developed some better prion diagnostic tests? So this was just a lab study in rodents where they infected the rodents with disease-causing prions, and they were able to detect disease-causing prions in the skin within 2 weeks, right? So I think they're hoping, well, maybe they could use the same approach in trying to diagnose prion diseases, say, in food animals like cattle, sheep, goats. And also, again, you guys, we really need to have a way to screen humans for it, right? Especially if we want to donate blood, donate organs. So it's just a link if you're interested. And then this, oh my gosh, we still don't have treatment for it. So, gosh, they're early on, folks, they thought in some animal models in the lab, they thought that maybe this anti-malarial drug, quinacrine, maybe slowed down the progression of the disease. And then another, it's fascinating, guys, chlorpromazine, an antipsychotic drug, again in a lab animal study. It looked like it slowed down the progression of the disease, but did not cure. And I'm not sure they've done these studies in humans, right, to see if these drugs might slow down the progression of the prion disease. And this, again, you guys, this is me being nerdy, so don't worry about it, but they're trying to see if they can develop antibodies that might protect against the spread of the disease-causing prions. So again, just kind of nerdy. Those of you that might be interested, just a paper where they're looking at antibodies, producing antibodies to see if they're administered passively, or if they can maybe, what they're trying to do is create a prion vaccine where you get vaccinated and you would make antibodies that could bind to the prions, right? Perhaps stop them from binding to other prions and causing the whole domino effect. And certainly, you guys, the prion diseases are, we're really interested in prion diseases, not just for them alone, but because they're a model for other protein misfolding diseases, which they call proteopathies. So you guys, some really serious other neurological diseases like Alzheimer's, Parkinson's, Lou Gehrig's disease, the amyotrophic lateral sclerosis. These are also ultimately fatal neurological diseases, and they all are the result of misfolding of proteins. So some people call these prion diseases, and I'm like, to me that's really confusing. And the reason is, folks, like, we know we have a gene for the good normal prion protein, and we know it's that protein that misfolds. So the proteins that misfold in Alzheimer's, Parkinson's, Lou Gehrig's disease, it's another protein. So to me, it's very confusing when people say, oh, these are prion diseases. I think what they mean is it's the misfolding protein model that's similar. right? And wouldn't it be awesome, you guys, if we found out a way to prevent the prions from misfolding? Maybe that would open the door to finding treatment for Alzheimer's and Parkinson's and Lou Gehrig's disease. And these are devastating diseases. Oh my gosh. Let me see, you guys, I think we did the scrapie one, right? Okay, yeah, we did the scrapie. And this is scrapie eradication. So in 2005, you guys, there's 105 infected flocks of goat or sheep. And I checked last week, the week before, and I think we are down to 3 or 4 flocks. So again, it's test and slaughter, devastating. But they are making progress. The only concern, you guys, is because with scrapie, we know that when the female goats or sheep give birth, the placenta and the placental fluids are chock full of the scrapie prions, and then that means that, you know, if they're giving birth out on the pasture becomes contaminated, and we know the prions can remain infectious for years and years. So, okay, don't want to think about that too much. All right.
So the ones without numbers, is that 0?
Yes, yeah. So that would mean that the flocks they had identified as of 2005 there, that was this one, you guys. So I'm presuming that in Arizona, New Mexico, Wyoming, for example, Washington, Nevada, no sheep or goat herds had been identified. The problem with the problem, you guys, is that sometimes if you don't look for it, you won't find it. And that's always the trouble with people, hospitals reporting, like cases. If they don't get reported, they show up as 0. So, I mean, I'm not trying to be cynical. It's just like, anyway. And again, you guys, just showing that, at least in the United States, the number of scrapie cases has gone way down because of these test and slaughter campaigns. And also, you guys, the reason that I wanted to show you, kind of remind you of the scrapie, is that we think the scrapie prions led to the emergence of the mad cow disease, the bovine spongiform encephalopathy. And you guys probably know this already, but we'll go through it. So to me, folks, this is a really powerful slide. It's by the World Health Organization, and it's trying to educate us, show us how animal products end up in substances that we might consume or use, and we might have no idea that these animal products are present. So the way the cartoon works, you guys, they're at if it's like a clock, they're at like, 8, 9, 10 o'clock. Those are the animals on the farms, and they've separated the ruminants, which are cattle, sheep, and goats, from the non-ruminants, the pigs, the poultry, and we would include fish if we're talking about food animals. So what they're showing there is the food animals, if they're healthy, they'll be slaughtered, right, and the meat will be used for human consumption. Parts of the carcass that people aren't going to eat, like skin, horns, hooves, will go for rendering, which is a cooking process. And then it's these rendered products that can end up in all kinds of things that we use, which we'll discuss. And sick animals can't be used as human food. Sick or dead animals on the farm, their bodies will be rendered. And so they think what happened in Britain is that goats and sheep that had died of scrapie or were incubating scrapie ended up being rendered, cooked, and then one of the products is this high protein supplement called blood and bone meal. And so that blood and bone meal was added to cattle feed, is a cheap source of protein. So you guys, would cooking destroy the disease-causing scrapie prions? No. So what happened was we humans fed the scrapie prions to cattle. Now cattle are not omnivores, they're not carnivores, they're herbivores. They don't eat animal, right? So that was our mistake, is that they're just plant eaters, right? But we were feeding them animal protein, and including the scrapie prions. So we think that's how the scrapie prion jumped into the cattle. The scrapie prion bound to the cattle, good prions caused them to misfold, and now, voila, we have a brand new disease of cattle bovine spongiform encephalopathy. Those cattle got sick, right, or died. Their bodies got rendered. So now we have BSE prions in our cattle feed, right? So it was like mortuary cannibalism, but we humans did it to the cattle, right? And so that's how it spread to the cattle. And then, folks, what happened eventually? It spread from cattle to humans, right, when we were eating the cattle tissues. Now, you might say, well, I'm just not going to eat beef. But this cartoon, you guys, shows us where those rendered animal products end up. And I'm always blown away by this. I know you can read, but this is just amazing to me. So let me read this. So, you guys, so salvage is rendering these products, right? So it says salvage. Knackers. Knackers is the name in Britain they give the people that do the salvage process with the sick dead animals. So it says knackers process non-edible animal remains, bone, skin, blood, feathers, rind, offal, as well as bodies of animals collected from farms. The waste is mixed and cooked using methods which are not always guaranteed to be effective against prions. And this makes me mad, you guys, because we know that the cooking processes do not destroy the disease-causing prion. It's like, just come out and say it. It's not safe. And then, you guys, various extracts. Be ready. Elastin, collagen beauty creams, glycerin and toothpastes, lipsticks, some soft drinks, keratin in shampoos. Did you have any idea? Tallow, animal fats. Omnipresent means it's everywhere. Omnipresent in foodstuffs, biscuits. And you guys, I think in Britain they call cookies biscuits, right? So it's like my favorite food group, cookies. Ice cream, stocks, soups, cooking oils, also used for making soap. Gelatin, from sweets to pâtés, desserts to medicines, even for the preparation of table wines. And you guys, as kids, did you love Jell-O gelatin? And then, folks, this is how we kind of kept passing the prions back to the cattle. So animal feeds, we'd add this blood and bone meal back to the animal feeds. So millions of tons of animal feed are produced each year with these rendered animal products in it. Now we have laws, you guys, where we aren't supposed to add the blood and bone meal to cattle or sheep or goat feed, right? But you can feed them to pigs and poultry and also to fish, for which it is presumed there is no risk, right? And I'm like, I would never trust a prion, you guys, never ever, yeah? And furthermore, you guys, the concern is if you have a feed mill, a mill that makes the animal feeds, they're like, okay, I'm going to run a batch of pig and poultry feed through, and I can use the protein supplements, right? I'm going to run it through my mill, and now, next week, I'll do cattle and sheep feed, right? Well, I'm like, how can you clean your machinery to make sure that any of those contaminating prions are not present when you run the cattle and sheep feed through? So, I mean, this is me being nerdy, you guys. I get it. But I'm just like, this isn't really all that safe. So what we think, folks, this is in German. It was the best cartoon I could get. I think this is German or Dutch, so you're not going crazy. So we think, you guys, that because the bodies of the scrapie-infected sheep and goats were used to make this protein supplement, the protein supplement was added to the cattle feed. So the scrapie prion jumped from sheep and goats into cattle, binding to the cattle prions, triggering them to misfold, and now we have the BSE, the mad cow prion, right? And so the mad cow disease, folks, I think it first appeared in Britain in the 80s. Right? And remember, the British Prime Minister says, no problem, you can eat British beef. But it wasn't even 10 years, you guys, before we start seeing CJD in young people. And again, how do we think the young people got it? From eating beef that had the BSE prion in it. And you might say, well, why young people? All right, well, so folks, when you're young, do you have a lot of money? I mean, if you're going to eat beef as a young person, I know I would get like the cheapest beef I could get, which was usually ground beef, really high fat content because it's cheap. So it's those ground meats that have tissues from different parts of the animal that are probably most likely to have prions. So you guys, in cattle, the tissues that are going to have really high concentration of prions are the brain, the spinal cord, the intestinal tract, and all the lymphoid tissue associated, and the tonsils, right? And so you might say, well, it seems to me, like when I eat beef, maybe I eat a steak, right? It seems like there shouldn't be any prions there. But remember, you guys, all muscle has neurons innervating it, sensory neurons, motor neurons. And furthermore, you guys, this is really disturbing, but it's important for us to know. In commercial packing plants, when the live cattle are brought in, big operations use bolt guns. And they use bolt guns to kill the cattle, so they put the bolt gun up to the forehead of the animal, they pull the trigger, and it drives a bolt into the brain under really high pressure. That's what kills the cattle. But they've shown you guys what they've done in studies. They use the bolt gun, and then they look for brain tissue. And the brain, because there's such force, right? Such force. Brain tissue gets spread down here and there and there. And the reason they're worried about that, right, is the brain is going to have a high concentration of prions. So maybe you're eating, like a cut, I don't know, from down here, and you might think, oh, it should be safe from prions, but it might be, there's contamination from this method of slaughter. You might say, well, I don't eat intestines, right? You might say that. But you guys, if we're making traditional sausages or hot dogs, what's the casing? It's intestines, right? So when you're buying hot dogs or sausage, right? I mean, anyway, it's interesting to see what the casing is. That's all I can say. And then you might say, well, why would I eat tonsil? Well, beef tongue is I think a pretty accepted form of beef. It's used like in delicatessen. And my fear would be that when they cut out the tongue, they could get the tonsil, and the tonsil is going to be potentially a rich source of prions. So you guys, so again, like they think one reason that young people got hit so hard is that young people weren't eating steaks that would have probably the lowest concentration of prions. The young people were eating the cuts of meat, ground meat, that were most likely to have the highest concentration of prions, and just total heartbreak. Okay. And so then, you guys, we finished the transmission cycle, scrapie prion to cattle. The BSE prion evolves. The BSE prion is eaten by humans, and the variant Creutzfeldt-Jacobs disease prion evolves, right? And if it weren't for us, you guys, this wouldn't have happened. This is what's so crazy making. So again, folks, there are feed bans now, so we aren't supposed to be feeding ruminant protein back to ruminants. Okay, so hopefully we're making some progress there. This still troubles me a little bit, you guys, because I think what they say now is if you're gonna slaughter cattle for human consumption, they have to be younger than 30 months, because usually cattle don't develop signs of mad cow disease after 30 months because of the long incubation period, right? But like we're told that is public to make us feel safer, but it's like, guys, just because the cow isn't demonstrating clinical signs and symptoms doesn't mean the cow couldn't be infected with prions, right? It takes 30 months often for the clinical signs and symptoms. I just think that it's not right for us to be told something to reassure us, and we're kind of being misled. That's the only thing that troubles me, right? So, and then you guys, here's another one that is, right. Okay, so we have our own North American prion disease. It's called chronic wasting disease, and it infects deer, elk, and moose. They've just added moose to the list. And folks, it evolved here in North America. The first cases were seen in deer in Colorado. And then notice, you guys, the red areas are parts of North America that are chronic wasting disease positive. So it's moved up north into Canada. It's moved west. It hasn't reached California. It's moved east. And it's moved south. And it's interesting, you guys, see how it seems to have jumped from, here's Illinois and Wisconsin, right? And then it seems like it jumped from Indiana over Ohio, from PA, and then it somehow got to New York and West Virginia. Like, how did that happen? Well, this, again, you guys, is through humans. Have you guys heard of game farms? Right, so game farms are where wild animals are raised, and then people come to hunt them. Now, you guys, now I have to let you know, my mom was raised in Colorado, and they hunted deer and elk. That was part of how their family survived. So I was raised on venison and elk. So I am not putting the hunting down. But game farm. Anyway, I just won't go there. Okay, so what do? So one game farm will ship deer, elk, I guess moose, to other game farms. So the reason the chronic wasting disease is jumping here is that these infected animals were transported by humans, right? So again, just kind of seeing how human behavior has played a role in the spread of these new prion diseases. So the transmission, they think, folks, is really similar to scrapie. So saliva is prion positive, blood's prion positive, placental fluids are prion positive. So where these infected animals are living and grazing, we presume, again, the pasture, the grasslands, the soil is getting contaminated with the chronic wasting disease prions, right? So the concern is, could chronic wasting disease jump into humans from like humans that eat venison, deer meat, or elk or moose? And I'm like, I don't see why that is not a possibility. Although we are told chronic wasting disease is not zoonotic, you guys, I would never eat meat from an animal that's chronic wasting disease prion positive. I just don't think that's a good idea. And furthermore, you guys, since the deer and elk and moose, they share grasslands, pasture, with our beef cattle. And so the other thought is, could the chronic wasting disease prion, just like scrapie, jump into cattle, right, if the cattle are eating the grasses contaminated? So it's definitely continuing to spread. There's some thoughts that some herds of deer are just going to be wiped out from chronic wasting disease. And the other big fear, you guys, is hunting a lot of people participate, and they hunt and they eat the carcass. And it's like, well, what if your deer or elk is incubating the chronic wasting disease, right? So they'll have check stands during hunting season. They'll have check stands. So if you're carrying like your carcass home, they'll try to inspect it. If it looks like the animal's emaciated, they'll confiscate it and test it. And also, folks, hunters can submit lymph nodes from the carcass and to have testing for prion disease, right? And my thought is, you guys, I truly would not eat meat that is from a CWD prion positive animal. I just don't think it's a good idea. A lot of hunters say, well, you know, it's okay. It's like a deer is like 200 pounds of venison. I'm not going to throw that away. But boy, you guys, I think we're playing with dynamite here. Yeah. Anyway, that's just me. And again, I come from a hunting family, but that's like I would not eat it. Okay. And this you guys. So this map of North America, so the gray, you guys, is chronic wasting disease in free-ranging populations, so wild herds. The darker is known CWD positives prior to 2000, so you can see it's spreading. Yellow is the game farms that are positive. Oh, depopulated. Sorry, you guys. Yellow is game farms that were chronic wasting disease positive, and they went in and wiped them all out. And red, you guys, is chronic wasting disease currently in game farms, right? So, gosh, a lot to think about. So, folks, there are TSCs in other animals. Cats can develop them. Mink can develop them. Squirrels can develop them. And you guys, you're like squirrels. Well, in some parts of the U.S., squirrel brains is kind of a delicacy. And you guys, I know, like, if you visit folks and they offer you food, the worst thing you can do is reject their food. I mean, it's so tough. But gosh, you guys, I would just really encourage you, whenever possible, try to avoid eating animal brains. And I'm not trying to be funny, right? Because animal brains, it's good nutrition, right? But with these prion diseases, oh my gosh, you guys. Oh. Yeah. And one thing I don't think I included here, you guys, is what about mad cow disease here in the United States? Okay, so the first case of mad cow disease that was diagnosed in the United States was in Washington, the state of Washington. And again, you guys, I have kind of a twisted sense of humor. So it was identified, and then the headlines were, oh, it was a Canadian cow. She was born in Canada. Blame Canada. And it was about the time that South Park movie. Anyway, you guys are probably too young for that. But anyway, it's like, blame Canada, blame Canada. But since then, there have been other cows diagnosed in the United States with mad cow disease. And they're always interested to know, well, was the cow or steer or bull born before the feed ban, meaning did they get infected by eating contaminated feed? Or were they born after the feed ban? And you guys, the latest case we had here in California, it's really troubling to me because it was a cow, I think it was a dairy cow here in California that developed mad cow disease. She was born after the feed ban, so they don't think it was contaminated feed. So you know what they decided? This is not comforting. They decided it was a sporadic form of BSE. Yeah, right? Right? And I'm like, oh my goodness. Yeah? And the government, right? No, the government. So folks, in Japan, every head of cattle that's going to go into the human food chain, every head is tested for BSE, right? Here in the United States, I think it's like maybe 0.1% of our cattle that are going to end up, being used for human food are tested. So that's a huge concern. But you guys, there's always a price, right? So if you go to Japan and you order beef, is it really expensive? Yeah. So, part of the reason it's so expensive is if they're really careful, right? Here in the United States, relatively speaking, is our beef pretty inexpensive? Like the conventionally raised, feedlot raised is pretty inexpensive. So you guys, again, as members of the public, it's like, right? I mean, to have safe food, we might have to end up paying more, right? And that's tough when people don't have enough money for healthy food already. So it's a dilemma, you guys. But just don't eat brain. That's all I need. Just don't eat brains. Okay. So you guys, these were some links just to papers on normal functions of prions. And like I said, you guys, these were so far over my head. I was like, oh my goodness. But I know a lot of you are like heavily into A&P, and you guys might have better understanding. So I just put some links here on the biological functions of prions because a lot of people are interested in those. And this was just I just cut some of what they said were the possible biological functions. I won't ask this on the exam, you guys, but people are always asking, well, what do prions do normally for us, right? And then is that it, or was there maybe one diagnosis? Yeah, and this was just another highly sophisticated diagnostic test that I could not explain to you. So I put the link here, you guys, because I know a lot of you are a lot sharper than I am. But with that, you guys, we're going to close the prion chapter. Okay. I mean, yeah. And I think one thing, folks, I just want to kind of underline this is that with some of our emerging infectious diseases, it's people that are kind of, I don't know how to say this, like interrupting natural cycles. Like, for example, you guys, cattle aren't meant to eat meat, right? And we force them to. So that was a human behavior that probably triggered the emergence of mad cow disease. So I think it's just really good for us as members of the public, right, and as biologists, for us to think about human interventions, quote unquote, and what some of the biological consequences might be. And I think this was a great one, you guys. If you have an herbivore, let them eat plants. They're not meant to eat meat, right? So anyway, all right, I better stop. So folks, we're going to leave prions unless, do you have any prion questions, you guys? And again, I know this much about prions, right? There's so many big question marks to me, but was there anything we discussed that I might be able to clarify for you? Yeah.
Is there any other way to test people for prions besides just waiting until they start having symptoms like
Well, my thought is if they can do tonsil biopsies on cattle and they can do rectal biopsies on sheep and goats, I know this sounds horrible. It's like, why can't we do the same on humans? So I think the rectal and tonsil biopsies, it's to detect the ingestive form, and maybe that's why we don't do it on humans. If 85% of Creutzfeldt-Jacobs disease in humans is sporadic, it might be tonsil biopsies and rectal biopsies won't work because it's not through ingestion.
So I guess why would you test for it if we can't do anything about it?
I know. I mean, I guess I would test for it if there was somebody that had lived in Britain, right? And I mean, because the idea is if you lived in Britain and you got infected through ingestion of beef, that is the ingestive form, and therefore possibly a tonsil biopsy or rectal biopsy might show the prion positive. But that's such a relatively low, like here in the States, it's such a low number of cases compared to the sporadic form. But I mean, but Nina, you're absolutely right. And maybe one of those later papers there, I mean, they're talking about diagnostic techniques that have to do with chemistry and physics. I couldn't even pronounce the name of the test. So they really are trying to use like higher order scientific diagnostic tests. I just don't know if they've made a lot of progress with it. They do in your handout, you guys, and it's not to make you crazy, but this was the Prion handout from a medical micro book. It was kind of more clinically oriented. And this isn't for you. You don't, obviously you don't have to read this, you guys, but they talk about more of the clinical signs and symptoms, and they talk about MRI abnormalities. What else? Yeah, they're talking about tonsil biopsies and variant Creutzfeldt-Jacobs. They're talking about, let me see here, MRI would be antemortem. They're talking about the neurological problems. So it looks like MRI, maybe you can pick up on some earlier signs, but again, you guys, and in the books that I had, there wasn't a lot of like really standout antemortem diagnostic tests. Yeah, but boy, we sure need them. Yeah. Okay. That wasn't very satisfactory, was it? Yeah, that's what I feel like on Prions. It's like I don't have many good answers, you know. Reassuring. Okay. Okay, I have 15 minutes, right? Okay. So folks, we're going to close Prions then and start the second to the last unit in the course. So we're starting to sprint for the finish line. Before we said it was a marathon, right? Now we're sprinting. So you guys, this next unit that will take 2 to 3 lectures is what we're calling medical microbiology. And the subtitle is How to Be a Good Pathogen. And there's actually some reason for this. Because folks, if we look at all the microbes in the world, do most of them cause disease? No, just the opposite. It's really unusual for a microorganism to cause harm to another organism. It's really unusual if you take into account the millions and millions of microbes in the world. So what we're going to do in this next unit is explore what is it a microbe has to do to become a good pathogen, to be able to cause harm to other organisms. Because, again, you guys, it's relatively rare. And what we're going to do, folks, is put the cart before the horse. We're going to talk about how the microbe is going to colonize us, outwit, suppress, defeat our immune system, how it's going to cause harm to us. And then, because this is the information, you guys, that to me is the most dense. And again, the handout today, the bacterial toxins, the virulence factors, it's a lot of stuff to memorize, and that's why I wanted to start on this part first. Once we finish this part A, then we're going to back up and look at what's called epidemiology, the study of how pathogens move through populations. And with part 2, we'll back up and we'll look at reservoirs. What are sources of the microbes? We'll look at transmission. What are all the different ways that a pathogen can leave its reservoir and then invade a new person, right? So again, we're kind of putting the cart before the horse. But so this one, you guys, medical micro intro, how to be a good pathogen. This is virulence factors and microbial mechanisms of pathogenicity. So folks, probably right off the bat, just to remind ourselves what we mean by a pathogen. So patho comes from pathos, which means suffering. And then if we add the suffix, you guys, gen, I think of something that makes something. So pathos, this is an organism or agent that can cause suffering. And the suffering may be caused by the pathogen invading another organism and upsetting normal homeostasis, causing tissue destruction. The suffering might be caused by the pathogen making a toxin, and then we ingest the toxin and it causes harm, right? So we say pathogenic, pathogenesis, these are all rooted in suffering, right? Okay. So in tackling this, folks, we want to back up historically and look at something you're taught from when you're, first your parents are talking to you. Your parents are probably educating you about germs, right? And your parents teach you how after you go to the bathroom you should wash your hands, right? Because you don't want germs on your hands. Your parents would probably teach you if you come across a dead animal, your parents are probably going to teach you, don't touch the dead animal, you can get germs, right? And they might infect you and cause disease. So you guys all probably almost intuitively know about the germ theory of disease, that germs, microbes can invade us or other organisms and cause harm. But you guys, this was amazing. For many centuries, people didn't believe in the germ theory of disease. People thought for many, many generations that if you developed disease, it was because you were a bad person or a witch or a wizard had cast a spell upon you, right? And even when work was done to prove that it's microorganisms that can cause disease, like say bubonic plague, people believed, well, you know what? The bubonic plague bacterium doesn't come from outside of you. It arises spontaneously within you because you're a bad person or because a witch or a wizard cast a spell upon you, right? So the germ theory of disease, you guys, again, you're like, well, of course everybody knows that. But the germ theory of disease states a microbe, a germ, can invade another organism and cause harm, right? And what's so important about the theory, you guys, is recognizing the microbe is coming from outside of you, right? And the reason that's important is that if we understand how the organism reaches you, that is, how is the organism transmitted, if we can block transmission, we can block disease. So this was just really important in the whole development of how to prevent and control infectious diseases. Okay, so there's two folks, you guys, that are kind of awarded the title of the provers of the germ theory of disease. And those two folks would be Louis Pasteur and a contemporary of his, Robert Koch. So Robert Koch was a German physician. Louis Pasteur was this phenomenal French scientist. So we'll start first, you guys, with Louis Pasteur, because a lot of people say he laid the foundation of the germ theory of disease. And it was really cool where he developed his ideas. And it was in his attempts to help the French wine industry, and then later I don't know how to pronounce it, guys, like silk producers, people that make silk, and they make the silk from silkworms that spin their cocoons, right? Okay. So Louis Pasteur described diseases of wine, and you're like, what the heck? Diseases of wine? What are you talking about? So historically, Pasteur is credited with saving the French wine industry. And the story is, you guys, when you're making wine, traditionally you use grapes, right? You get the grape juice, and then through fermentation, you're supposed to make alcohol from the grape sugars, right? But what was happening in France is the wine producers were going crazy because the wine was ending up tasting sour. And you guys, remember, we detect acids as sour, right? So the grape juice was being fermented into acids, not alcohol. So everybody's panicked. They didn't know what the heck was going on. And what Pasteur discovered was that it was contaminating bacteria that were carrying out, like fermentation resulting in acids. That's why the wine tasted sour. So it was like those bacteria were pathogens of the wine, right? Diseases of the wine. So do you guys have any idea how he solved their problem? It was brilliant. Pasteurization, right? He said, here's what you do. You take your grape juice, you heat it to kill those contaminating bacteria, those pathogens. You heat the wine to kill the pathogens, and then you inoculate with what? Saccharomyces cerevisiae, right? And voila, that's the only French word I know. He saved the French wine industry. But you guys, here he's developing this concept of a pathogen coming from the outside causing disease, right? Even though you think fine, right? But then he applied the same concept, you guys, because the silk producers, their silkworms were getting sick, right? And if the silkworms aren't alive and make their little cocoons, that's where the silk comes from, right? And so Pasteur, kind of using this germ theory of disease, he goes, I wonder if the silkworms are getting infected with a microbial pathogen. And he discovered that was true, right? And you guys, I haven't read enough to see how he solved that one, but he showed the silkworms were getting infected, and that's why they were dying. So he was able to help the silk producers save their livelihoods. So again, you guys, he had this germ theory of disease in mind. But you guys, one disadvantage Pasteur in his lab had is they were only, when they were working with their bacterial pathogen suspects, their lab only used broths, liquid medium. And you guys, what's the problem with broths? Yeah, it's hard to look at them and know if they're pure or not, right? And we're going to see, to prove the germ theory of disease, folks, you have to have pure cultures. And because Pasteur's lab was working with broth cultures, liquid cultures, he could never guarantee he had a pure culture. But Koch's lab, Robert Koch, his competitor over in Germany, one of Robert Koch's colleagues was discussing this problem of how to develop a solid medium that microbes wouldn't digest, a solid medium that would remain solid on hot days. And it was one of Koch's colleagues' wife suggested try using agar. And agar in the days, you guys, was used to make jellies, right? And she said, well, try using agar. And they did, and it was miraculous, right? So we still use agar today, you guys, to make our solid media. So Koch's lab, they had a huge advantage, folks. They had solid media. They were using agar, like agar slants and agar plates. And we know, you guys, when we grow our microbial cultures on agar plates, if we get isolated colonies, just by inspecting with the naked eye, we can get some sense of whether the culture is pure or not. How would an agar plate appear, you guys, if you had a pure culture? The colonies should look the same, right? And how would your agar plate appear, you guys, if you have a mixed culture, sorry. Different types of colonies, right? And you guys do remember an isolated colony is pure culture, right? All the cells in the colony, 10 to the 8, 10 to the 9 cells, are all descended from a single cell. So they're genetically identical, right? And if we have more agar plates, we could use aseptic technique and transfer cells from that isolated colony to a new plate, and voila. In 24 hours, we have pure culture. And then we can run our diagnostic tests. We can run tests to see if this pathogen can cause a particular disease, right? So these guys had that huge advantage. They could guarantee they had pure cultures. And I think it was because of that, you guys, that Robert Koch kind of sort of won the race to prove the germ theory of disease, although both these folks, I think, played really important roles. So you guys, again, Robert Koch and his colleagues, they developed 4 prerequisites, 4 steps to prove a specific pathogen causes a specific infectious disease. And these 4 steps, you guys, these 4 prerequisites, what do you think we call them? Koch's postulates, right? And so Koch developed this test to prove a specific pathogen causes a specific infectious disease. And you guys, it was in developing his postulates then he proved the germ theory of disease. He actually had the experimental protocol to prove a specific microbe can cause a specific infectious disease. So folks, what I probably do in the lecture exam 3 is ask you, like give you a multiple choice with all these different steps and say, I could word it one of 2 ways. I could say which one is part of Koch's 4 postulates or which one is not a part of Koch's 4 postulates. So you guys, let me just run through them really quickly, okay? So folks, the first of Koch's postulates is, let's say you guys, the experiment is, I want to prove that microbe X causes infectious disease X, right? We'll just keep it generic. So the first postulate is that you must be able to identify the suspect microbe X from all cases of disease X, right? And then the second part, you guys, this is where the pure culture becomes really important. you have to isolate in pure culture the suspect microbe X from all cases of infectious disease X. So see how pure culture is so important here? Okay. And so there you guys, you can see the street plates that are pure culture. And then step 3, you guys, this is the ethical moral problem with Koch's postulates. You then must take your pure culture of your suspect pathogen and inject healthy animals with the suspect pathogen, right? And now your healthy animals have to do what? Get sick or die, right? And then the 4th step is from those sickened or dead test animals, what must you do? Re-isolate the suspect pathogen in pure culture, right? So you guys, this is big moral ethical issues and using, purposely infecting animals. That's a moral ethical issue. There's some pathogens, you guys, where humans are the only host, right? And it's like, well, that's not going to work, right? Can't perform Koch's postulates on a pathogen that only has humans, especially if it's a really lethal microbe. And you guys, there's another problem with Koch's postulates in that, what about viruses, right? Can you grow viruses in pure culture? No, right? Because they have to grow inside another cell. So, I mean, there's all these little like potential problems with Koch's postulates. But you guys, they're still kind of the foundation. They're the foundation that providing the steps, the test, by which we still will try to attempt, if we discover a new pathogen, try to verify that it causes a specific infectious disease. Just acknowledging, you guys, a lot of times we can't fulfill all of Koch's postulates. Okay. But folks, again, do you see where pure culture was so important here? Yeah, absolutely crucial. And then, folks, we just have a few minutes remaining. But again, just kind of warming up to these virulence factors. We want to compare, you guys. We know, let me ask you a question, you guys. Is a cold virus a pathogen? Is a cold virus a pathogen? Yeah. Can it invade you and make you suffer? I mean, it's not horrible, but stuffy nose and sneezing and coughing, right? So, you guys, so we call the cold virus is a pathogen, right? And then, folks, is the Ebola virus a pathogen? Yeah. And folks, let's just do a mind game. Let's do a mind game and pretend that you must be infected with one of these viruses. No choice. Which one do you want to be infected with? The cold virus. I agree. Why? It's not likely to cause really serious harm, right? It's not going to kill you, right? But what about Ebola virus? Yeah, some strains, you guys, have 90% mortality rates, right? So you guys, so even though they're both pathogens, we need a way to express how much damage they can cause, yeah? And so the term we use to describe how much damage they can cause, maybe you could say how dangerous they are, is virulence, yeah? Okay, so you guys, if virulence is a measure of how much damage the microbe can do, maybe the potential to kill you, would you describe the cold virus as high virulence or low virulence? Low virulence, good. In contrast, folks, how would you describe Ebola virus? High virulence or low virulence? Really high virulence, yeah? And folks, what we'll do then is on, today's Wednesday, right? On Monday, we'll start with this slide, and we'll talk about how we can calculate what's called infectious dose 50 and lethal dose 50. Infectious dose 50, you guys, is how many microbes it takes to infect 50% of a test population of animals. Lethal dose 50 is how many microbes it takes to kill 50% of a test population of organisms. And you guys, the key here, just so you can think about it, there's an inverse relationship between infectious dose 50, LD 50, and virulence, meaning as infectious dose 50, LD 50 goes down, what happens to virulence? It goes up, right? And as infectious dose 50, LD 50 goes up, what's happening to virulence? It's going down. Okay, so we'll just leave it with that, you guys, and then we'll explain why that should be. And then we'll look at some of the pathogens. We'll compare the virulence of some of the pathogens we have here in California. Okay? So you guys take good care of No, I'm not going to see you guys unless I see you at open labs. You guys have a good weekend, okay?
I kind of like. I hate being so windy.